we have a very, very special speaker, Professor Himender Bharti. I know Himender for the last like 6-7 years and have met him in a couple of meetings. And every other week, he finds a new species and posts it on the Facebook. And so he's doing this wonderful job, in the field of taxonomy in our country. Himender currently is the director of Center for Restoration of Ecosystems of Punjab, established at Punjabi University, Patiala, and former head of the Department of Geology and Environmental Sciences there. He's an evolutionary biologist with a keen interest in systematics, phylogeography, speciation pattern, phylogenetics, ecology, and conservation biology. He's widely traveled and has the distinction of visiting Natural History Museum London and other natural history museums in Southeast Asia. Professor Bharti has been nominated as Fellow of prestigious Royal Entomological Society London and has also been conferred with the Distinguished Taxonomist Award by T.C. Narendra Trust for Animal Taxonomy. And as a naturalist, he has been visiting Himalayan regions, Western Ghats, and tropical forests of Southeast Asia. In his quest of the natural world, he has been exploring the intricacies of wonderful world of ants. And he is credited with the discovery of more than 150 new species of ants, which include red list species as well. It's a wonderful job, Himender, you have done this. He is instrumental in building a comprehensive database of Indian ants. He has a wonderful web page. Just give some time. We'll find lots of interesting stuff there. Dr. Bharti has published more than 150 research papers till date. His ventures have throughout been sponsored by national funding agencies like Department of Science and Technology, New Delhi, Ministry of Environment, Forest and Climate Change, and the Indian Council for Agricultural Research. As an internationally accomplished scientist, he is involved in Discovery Life Project at Harvard University, Global Ant Project, Pharmacy Barcode of Life, Global Project on Ant Genomics, Encyclopedia of Life, Ant Wiki, and Ant Web Project, California Academy of Sciences. For red listing of Asian ants, Dr. Bharti is also involved with IUCN, which is International Union for Conservation of Nature and Natural Resources. At present, he happens to be the president of ANET, which means International Network for Study of Ants, and IUSSI, which means International Union for Study of Social Insects, Indian chapter. He's on the editorial board of international journals like Asian Myrmecology, Sociobiology, Current Research in Insect Science, and is the chief editor of journal Halters, which he initiated recently. And besides being reviewer for many international journals, his lectures, talks for popularization of science has inspired many young minds to pursue the passion in natural history, and he envisions biology as integrative organismal biology, and that's why, Himender, we have invited you here. So very welcome, please.
So maybe next 30 or 35 minutes, I'll talk around something which is what we call taxonomy, how it has progressed, how we are standing currently now, and what we should do in current scenario. So this is something stuff, how it has progressed, how it has developed over course of time, and how the changes have occurred, and how we should perceive about taxonomy in present moment. Okay, so that's why the title of talk is Growth of Linnaean Thought, Impact of Common Descent, and Current Understanding. So I'll start with, say, bit of historical backdrop, how it has progressed, then principle of common descent, that is origin, and what was the impact of this common descent on species, then traditional taxonomy, then how we should think in terms of species delimitation, because whenever we talk about taxonomy, the major issue is species delimitation. So that is the major concern. And obviously now we have concerns regarding phylogenetics, that phylogenetics has invaded taxonomy also. So there is a lot of revolution in terms of molecular biology. So given this backdrop, what I feel is there is a lack of proper understanding that how we should go for it. Now this was, everybody has been talking about Carl von Linnaeus. So 17th century was a deluge of information regarding specimens. And this was the man who initiated binomial nomenclature. He compiled the whole information, whatever was available at that point of time, in a tabulated form. So although we call it as scientific classification, so that was the scenario in 17th century. In 1758, he published a book, all of you know, Systema Naturae. Then he gave a ladder type classification system. That is what we call as hierarchical classification system. And now we call him as father of modern taxonomy, because he was the first one who could compile the whole information, what was gathered at that point of time, in a sort of, you can say, hierarchical order. So this is a typical hierarchical order which you study right from your school days. That is, when we talk in terms of hierarchy, we talk of these 7 obligate categories. So when I say obligate, it means whenever you classify an organism, these 7 should be named. That is, they should be, the organism should be placed in that sense. For example, if we are working in university or college systems, if students do not write this type of classification, we say that this is not complete classification. Everything should be mentioned. However, the concern is that this whole classification, although till date we follow this classification or this hierarchical order, the concern is that sometimes it doesn't reflect natural relationships. So when I say natural relationships, I'll come to that later. So right from kingdom up to species, that is a type of order which we generate. If you move downwards, right from kingdom to species, you find similarities, they do increase. So if you compare two closely allied species, you will find minimal differences among them. And if you go up, you will find major differences among different. And that was in 1758. Later, all of you know that Darwinian revolution was there in 1859 when Darwin published his book Origin of Species by means of natural selection, and the same theory, the same thought was brought by Alfred Russel Wallace also. So I call it as Darwin and Wallace theory of natural selection. So at that point of time, there was a thinking that there should be some idea of common descent, that how species have originated, how they have diversified, and there might be something, there should be some common ancestor from where they originate. So when we simply classify, we do not look up to relationships from where they have originated or how they are related to each other. I call it as unnatural system of classification. So what is natural system of classification? When we look at different relationships. So this was the tree which was published in Darwin's book, and there simply he wrote, I think this could be the possible view that multiple branches are there from common ancestor and they have branched that way. So that is the idea of common descent. And later on, it was emphasized by embryologist or developmental biologist Ernest Haeckel also. So that was the time and with this impact, that given Linnaean backdrop, what has happened, that there was influence of principle of descent with modifications on traditional taxonomy. Because what Linnaeus did, that was arbitrary classification. If I say arbitrary, that is based on similarities. So I'll come to that later. When we say based on similarities, that you look at organisms in terms of similarities, and based on those similarities, you make clusters, and based on those clusters, you start classifying or making them in arranging them in a particular order. So, but there was no idea regarding from where they have originated. What is the reason? Because that was 17th century. Now we have moved to 18th century. So that was the influence of principle of descent with modifications. Then we started looking for scientific explanation for existence of that taxa. That taxa means group of organisms, that from where they have originated, how they have come up, how they are interrelated to one another. And eventually that led to changed understanding of structure of taxonomic relationships, that how we should look. And by doing so, nested, which are related to each other or nested in each other or mutually exclusive relationships became primary significance. And then partially overlapping, they were relegated to secondary status. So the whole crux is that people started thinking in terms of scientific explanation about existence of taxa. Now, this is the major issue even now. When we define species, we do not know what should be the exact definition. that how we should demarcate that this particular population has differentiated to such an extent from some other population so that we call it as a different species. We have multiple definitions. I'll come to that later in part of this presentation. So basically, there are clusters of populations which we think if they resemble each other or there are some characteristics based on which we classify. Because when we say they are reproductively isolated, it is very difficult to judge reproductive isolation in field. Because now this is the major impact which Ernest Mayr had at that time, that when he defined that what is species in 40s, till today we follow the same definition. That is, he said that when existing populations, they become separated due to some geographical barrier, what we call as allopatric mode of speciation. So he championed allopatric mode of speciation, the reason being, whenever populations are separated from each other or they are disjunct from each other, the concern is that we have to prevent gene flow to such an extent for a long period of time so that some of the genes or some of the loci become differentiated. And whenever they come in contact with each other, we do not expect any kind of hybridization or any kind of reproduction or mating among them. So this was the basic idea, but the concern was that how to judge reproductive isolation. That is a major concern. So he said group of interbreeding populations which are reproductively isolated from such other groups, they should be considered as valid species. So this we call as biological species concept. That is biological definition of species, that how species should be defined. So being in systematics or taxonomy, practicing taxonomy or systematics without understanding how speciation occurs or what are the factors which are responsible for species, it becomes very difficult and challenging. Then it didn't have much effect on Linnaean hierarchy because groups nested within group structure mirrored the taxonomic structure implied by the principle of descent. Till today, even in schools and university level, we study the same pattern. All of you know Pisces, aves, amphibians, birds and aves and birds and mammals and reptiles. We still follow the Linnaean type of classification because at some point of time it holds true. Although it was based on similarities, but at some point of time it holds true. Then later in early 1900, there was new systematics. Julian Huxley coined this term. Then Mayr, because at that point of time, Darwinian theory was elaborated in terms of modern evolutionary synthesis. Doswensky, then Huxley, then Mayr, all these Stebbins, they combined together that modern evolutionary synthesis, that how evolution occurs, what is the role of species, then reformulation of species concept, and species were considered as population, not as group of similar organisms. So this is important. They were considered as populations, but not as groups of similar organisms, and they were considered as basic evolutionary unit, that where lineages, they start differentiating, or the populations, they start differentiating, or the population have some sort of standing genetic variation based on which they can deviate or they can diverge given different type of selection pressures, maybe in terms of geographical isolation, maybe in terms of ecology or some other factors. And then there was a rise of another kind of systematics, which we call cladistics or phylogenetic systematics. So it's not new, although it has been replaced by DNA barcoding and other kind of stuff. But this all started in 60s. Now, these higher taxa, which was sort of assemblage of similar species in Linnaean system, which second slide, it was their hierarchical classification, kingdom, phylum, like that. So that is a sort of clustering of different taxa in higher order based on similarities. So it was replaced with clades. Clade means branch. So it was replaced with clades. And then there was an idea which was floated by phylogenetic systematics, monophyletic. That is means units of exclusive common ancestry. That those groups, those species, those populations, those lineages which have a common ancestor, they should be considered as monophyletic. Monophyletic is not important. Important, or the significance of this term is that monophily means those groups should be considered natural. how they have originated in nature, how they have diversified in nature, and the way they are interrelated to each other, that is known as natural. That is, we are looking for natural relationships. The group which is called being natural, that should be monophyletic. It should have a common ancestor based on principle of descent. And phenotypic similarity, as I said earlier, in Linnaean system, that was not the basis. And I'll come to this later, elimination of paraphyletic and polyphyletic. So given this backdrop, now the challenge is how to define species and how to delimit species. That is the primary task of a taxonomist, or primary task of a systematist, that how species should be defined and how some population should be delimited or categorized as a valid species as compared to the second population. So that is the major challenge. So as Priyan has also, Harshan also, that we know what is the purpose of taxonomy. Extinction rates are quite high now. There's climate change. They are window to natural history. We require conservation. Then there is maintenance and resilience of ecosystems. So this is a kind of significance. And E.O. Wilson, all of you might have heard, he said that we should set aside half Earth for rest of the animals or rest of the species so that we can survive together. So he gave a half Earth project. Now, starting with, first of all, there was a lot of influence of morphology on taxonomy, because most of the time, our descriptions are based on morphology. If we look for specimens after my talk, we are looking for morphology. And we do assume that morphology is a typical expression of genes. So when we do expect that morphology or phenotype is a typical expression of genes, we do assume that this is a reflection of what is inside. So when we talk of morphology, earlier there was a taxonomy entirely based on morphology. Nowadays, what we call it as morphotaxonomy. Earlier, if you used to describe species, say 10 or 20 years earlier, we would say it is a taxonomy. But now they say it is a morphotaxonomy because your observations are based on external appearance or phenotype, which is known as morphology. So what we do in morphotaxonomy, that's why I have put a title morphotaxonomy. We go for certain features, that is based on certain features which we can see, which we can visualize. Then we go for images, we go for measurements, indices. Then now the question is, we can't judge reproductive isolation based on morphology. What we can do is look for traits. We look for those traits, we look for those features which we assume help in reproductive isolation, that based on these features, these two populations are different from each other so that they may not interbreed. These structures, these behavioral changes, these morphological changes or ecological changes are basically reflection of their differentiation. Then in insects also, we look for genitalia because we consider that genitalic structures play a major role in reproduction. So that's why if there are certain differences in genitalia, so there could be not a possible sort of mating among different insect groups or different insect species, then we go for images. This is started in 2000. Harvard University started this. The specimen on which we describe some species, we call it as holotype. So it was very difficult to get those specimens from international museums because most of our stuff is lying in Natural History Museum London, in Geneva, and other museums. So they started with this. They call it as cybertype. So this is a high resolution image from where you can compare your specimens easily, whether it is the same or it is different. Then these kind of, you can say, are available. Then we have web resources based on morphotaxonomy, comparisons, distributions, and I call it as cyber taxonomy because it is entirely based on informatics. And this is a kind of like, see, if this is one of the red list species, as Vash has pointed out, which I described in 2012 from Himalaya. So if you go to website, you will find this species images, the place from where it has been collected, then you will find the complete description. So this whole description is based on morphology, that how this species is different from other species. Then we have citizen science. Now we are coming up with the kind of stuff that we can easily identify species based on field guides, everything. It's good that we can have some awareness of biodiversity, but it is not the primary duty of a scientist or a researcher to go for such a work, because you have complete insight what you're doing and why you are doing. So that is more important. And then relevance and conservation in present text. So coming after morphotaxonomy, there was another school of thought, phonetics. So they thought it is better to look for similar characters, make a pool or data of those characters in terms of number of similar characters, and then compare. For example, if there are 50 characters which are similar with population A and B, or there are 30, 40 based on that kind of data, you can think in terms of that. That is known as numerical taxonomy, or since it is based on phenotype, external similarity, it was called phonetics at that point of time. And these two scientists in 63, Sokal and Sneath, they gave this idea. So, but not much useful, because again, it is based on overall similarities. Then this person, Willy Hennings, in 60s, he wrote a book, Phylogenetics, that was in German. And this was I would say this was a revolutionary idea which he put forth in taxonomy. That is, he said that we should focus on those characters which are derived. Which are derived, it means at some point of time, they are not ancestral characters, they are derived in those two populations. If they are derived in those two populations, it means those two populations have originated from common ancestor. So he gave this term cladistics, but the major issue was his book was not translated into English, because whole idea was in German. So it was in 1979 when the book was translated and it saw the light of the day. But the concern was, again, the translation was not appropriate. So he gave the concept that if you look for derived characters, they are known as synapomorphies. Shared apomorphy means single derived character. If it is shared by two, we call it as synapomorphy. And that is character state because it originated in immediate common ancestor. That is, it is not present in remote ancestor. In some of the immediate common ancestor, it has appeared, and suddenly it is appearing in the descendants. If it is so, we call it as monophyletic. So he gave the idea of monophyly, and he said that only those groups should be considered natural. Those groups should be given significance in systematics or taxonomy, which are related to each other on the basis of derived characters or synapomorphy. So this idea was monophyly. Whatever, if there is a resemblance based on certain similarities or convergences, he called them polyphyly. Polyphyly, like algae we know or algae we know, they are polyphyletic group. Because when we compare different groups together, it is basically we compare or we look at them based on their similarities. So that is, if some kind of origin is there, that if we are looking at similarities, this is known as polyphyletic origin. And if we are looking at a group based on certain remote ancestor similarities, that is known as paraphyletic. If certain resemblances are there based on, say, similar selection pressures or environmental conditions, that is polyphyletic. If certain resemblances are there based on similarities which have come from remote ancestor, that is paraphyletic. So the meaning of whole story is that, according to Henning, those groups which are polyphyletic or those are paraphyletic should not be recognized as natural. Only monophyletic groups should be recognized as natural. So when we think in terms of descriptive taxonomy based on morphotaxonomy, and then suddenly jumping into phylogenetics without understanding the whole concept, it becomes very difficult. So this whole scenario should be incorporated into the whole story, that how we should go for it. For example, this is a typical scenario. You know, in case of cactus, spines are modified leaves. In euphorbia, they are modified shoots. So if I say they are the same, a person may say they are polyphyletic, because origin of both the traits is entirely different, although they resemble each other. They resemble each other, but we know the origin is different. So I would say they are polyphyletic. They do not belong to same ancestor. They are not monophyletic. Now, this typical scenario, everybody studies this, graduation, post-graduation, PhD. We know reptiles. We consider reptiles as a distinct taxon, as a distinct assemblage, which is being worthy of given a name or assigned a name. Like Pisces, we know fishes, then amphibians, then we know aves, then reptiles, then we have these mammals. But if we go by cladistics, Reptile as a group is paraphyletic. It is not natural group. If we have to consider it as a natural group, either we should include birds into it, or we should remove crocodiles out of it. So that kind of, because four-chambered heart, three-chambered heart, there are a lot of problems in that. So this is typically based on certain similarities, although we follow this stuff, but in terms of cladistics, we do not agree with this. We consider it as paraphyletic group. So this was one of my papers, which created a lot of problem all over the world, published in Insects OCX, because in ants we have social parasites. So social parasite means when, ants are social organisms. So social parasite is a type of, you can say, species or type of organism, a type of population which invades a host and then starts living with it. Then in morphotaxonomy, it is generally considered that that parasite has originated from the host. Parasite has originated from the host, and morphologically parasite resembles host on the basis of certain similarities that differs from host on the basis of certain apomorphies or certain derived characters. So now this is an interesting stuff, although it is a bit difficult to comprehend. Let me make it very simple. That on one hand, we are thinking in terms of Linnaean taxonomy. That is, we are still following the same idea, species, genus, species, similar names. Then we are following the same obligate categories, which I said 7 obligate categories. On other hand, we are looking at natural relationships. And when we said that this parasite is a different species, then they went for phylogeny based on nuclear genes. 11 nuclear genes, they did a phylogeny on parasites. And then they said to make this group monophyletic, the species which we have raised in a different genus as a different species, it should be included in the same genus. So that means, on other hand, what they are doing, they are executing that this should only then it can be considered monophyletic if it is included broadly in that group. But on other hand, they are banning it on the basis of paraphyly. They are saying that similarities are based on certain ancestors. So we are doing two conflicting things or two contradictory things. First, we are doing Linnaean taxonomy, and other, we are looking at a concept which is entirely different based on origin. Now, when this cladistics or Willy Henning's concept was introduced of monophily, the biological taxon or the groups or assemblages that transform the Linnaean thought, that how we should go for it. That is, there should be some criteria which should be objective. When I say objective, if you go by the definition of biological species concept, you simply say group of interbreeding populations or potentially interbreeding population. This is important. When we say group of interbreeding populations or potentially interbreeding populations, which are reproductively isolated from such other groups. When I say potentially interbreeding, it means I'm assuming something. And when I'm assuming something, obviously it is based on certain traits or certain features. And when it is based on certain features, it means everything depends upon my subjective thought. There's no objectivity in that. So that kind of species concept should be there, which should be objective. And in Linnaean thought, these are basically arbitrary ranks, and species are unitary populations or population lineages, where supra-specific taxa were groups of such lineages. That is, supra-specific means when we move above species. Species with similar features, we call them genus or genera. Then we go up family, order based on certain features, we start combining those. For example, if you enter a library, you say that is a section sciences, that is a broader. Then you go inside physical sciences, chemical sciences, then go inside biological sciences. Then in biological sciences, ultimately you go botany, zoology, microbiology. So that is a kind of stuff. If you start from the scratch, you end up doing sciences and humanities. So that is a kind of hierarchy which you are generating. Now, morphology had a lot of constraints, as I said earlier. Convergences or homoplasy. You know already that some of the features which organisms based on which organisms resemble each other, they could be based on convergence or homoplasy, and some of the species complexes also which are difficult to handle. That species, they appear as similar, but they may be different also. They may be reproductively isolated cryptic species, large fraction of biodiversity, that those has been erroneously named under one category or one species. Then morphological stasis. You might have heard of Stephen Jay Gould. He gave the idea of punctuated equilibrium. So he said that species, they remain in a period of stasis for a long period of time, and suddenly there is differentiation. So at one point of time, he challenged Darwin's theory. When we say gradualism is there, that slowly changes accumulate or slowly changes do occur. But he said that for long period of time, species, they remain the same, the populations, they remain the same, and suddenly there is diversification. So that is basically morphological stasis. His idea was entirely based on fossils, because in fossils, he couldn't get the whole series. If we couldn't get the whole series, then suddenly if you get intermittent or intermediate fossils, you may find this fossil has some features, then suddenly you find a fossil which has different features. So that kind of idea was there. So this is known as morphological stasis, that if you look at like fish coelacanth, we call it as living fossil. For a long period of time, we called it as living fossils. Suddenly they find the genome and they find that, as Harshad has pointed out, that a lot of changes in the genome. What we are looking at morphology. And based on morphology, we are saying it's a living fossil because there are no changes in morphology. So that is basically known as morphological stasis. So as taxonomists, based on morphology or based on morphotaxonomy, most of the problems which are created, two types of schools are there. Lumping, that is whatever is existing earlier. If I say this is a new species, someone comes after me, he says, this is not new. This is an old species. So that is synonymization. That is, he would say that he or she would say that it should be placed under already described species. It's not new. That is lumping or splitting. If I say this population is different from other population, I may say this is a new species. So based on thinking or the type of thought we carry and type of comprehension we do have, based on that kind of comprehension, we fall in these two categories, majority. That is either splitters or lumpers. So where does traditional taxonomy stand now? Given this backdrop which I have talked about now, where do we stand now? Now, he said that there should be ecological. Now, from morphology, we have moved to there should be some geographical information, there should be some ecological information, there should be some behavioral information, there should be some molecular information, all should be integrated together, so that we can say isolated populations have differentiated to such a level that we consider them as different species. And this is another example where we can go, for example, this is based on scanning electron microscopy. You go for certain features and start comparing different populations, whether they are different. This is a typical scenario, ecological information based on ecology, then cytotaxonomy based on chromosomes, like Subhash and Harshit are doing cuticular hydrocarbons. People are going after them also, that they can also be used as species delimiting tools. So these are the kind of examples which I'm telling you, that these tools are also being used with passage of time. Then geographical isolation. If they live in different areas, we assume they are exposed to different selection pressures. And if they are exposed to different selection pressures, it means there might be some sort of differentiation in genome as well. Then 1994, there was a revolution in the sense that he said that we should go, all of you might have studied molecular markers. That's a separate topic in graduation or post-graduation, molecular markers. So there was a deluge that molecular markers should be used, protein immunology, rapid and amplified polymorphic DNA, relative fragment length polymorphism, AFLP. Then we go for microsatellites. Now we have come at the level of genes, what we should do now. So there was the information which they put together that this should also be taken into account while thinking in terms of differentiation. Then in 2003, there was a big leap. Paul Hubbard, he came up with DNA barcoding, that everything should be barcoded, or there is a segment of DNA lying in mitochondria, which is basically useful for identifying different species or delimiting different populations as species, or looking for those. Then there was International Barcode of Life project, which was initiated in 2004. And the basis, we already know, the typical basis was that there is single mitochondrial DNA haploid, it evolves rapidly because there is an inefficient repair mechanism. It evolves at a faster rate and it is transmitted predominantly through maternal lines. So there is no scope of recombination, because if it is through both parents, then there are chances of recombination. Then from functional perspective, it has 37 genes, but from phylogenetic perspective, it is a genealogical unit with numerous alleles. So out of that, cytochrome oxidase was chosen. But the problem is that, again, we need to understand before taking this into, you can say, action, or taking this, thinking in terms of using this. There are a lot of problems. First is hybridization. Second is introgression. And third is incomplete lineage sorting. Now it depends what kind of genes or what kind of loci are you using for this species delimitation or generating natural relationships. That is more important. It has become pretty common over the last 5 or 6 years that a lot of hybridization has been reported in different species. Although they have been primarily classified as different species, but there is a lot of hybridization. If there is hybridization and hybrids do survive, it means they interbreed. And hybrids are equally fit, it means they interbreed, and there is no such level of differentiation that they do not interbreed or there are no hybrids. Like you might have heard, Neanderthals, they introgressed their genome into Homo sapiens 40,000 years ago. So that is a kind of introgression, like a gene for COVID-19, we are susceptible to that. That has been introgressed from Neanderthals. So that kind of situation is there. So saying that species are reproductively isolated has no meaning, because still we can go for hybridization. Now look at this and tell me what you find in this. These three: human, chimpanzee, gorilla. 4 different markers have been used, 4 different genes have been taken into account, and the outer black is basically species tree. That as we see them now, we see humans are different species, chimpanzee different, and gorilla different. Now we have 4 different genes. Can you find any difference among these 4 pictures? Here, this gene clearly shows that humans are related to chimpanzee. Here, then over here, splitting is entirely different, and when you go here, it is entirely different. It means when these populations, they differentiated on a different path from a common ancestor, it means the genes in question did not follow the same pattern. They did not diversify at the same rate as does the populations do. First case, it is fine. The gene in question is okay. Because from here, human and chimpanzee, this gene clearly shows they are related to each other. Because here, it is originating then from here. It has gone deep. Here, it is entirely different. Now, if you look at this gene, it will show human and gorilla are related. So these 3 would say, I would say, incomplete lineage sorting. That means these lineages have not been completely sorted based on genes, although we know they are different species. So to keep in mind, gene trees and species trees are entirely different. Huh? Like this is a gene in question, right? This is the speciation event. Speciation event when populations have diverged on different paths. Then gene, this is ancestral copy. It has gone to gorilla. Here, it has again undergone modification or duplication. It has gone to human and chimpanzee. If I go back, or we call it as coalescence. If I go back or track back the history, I will find that when these 2 populations or these 2 species, they got diversified, gene also got diversified at the same time. If I'm using this gene for phylogenetic analysis or species delimitation, my results would be okay. Now look at the rest of the 3. Common ancestor, but they are not synchronized. There is no synchrony between gene separation and species separation. And third, now this has come over And if we look at this, we know they are different. Now, what will be our conclusion? Yes, and over here, chimpanzee and gorilla are common ancestors. So this kind of understanding should be there. Now look at this. This is we call it as reciprocal monophily. It means that don't get confused by this picture. This is a common ancestral population. From this common ancestral population or common ancestral, you can say lineages, these two populations have diverged. Now, when these two populations have diverged, it means both the populations have diverged to such an extent that there is a differentiation in those populations. They do not resemble each other. It means they are reciprocally monophyletic to each other. If reciprocal monophily doesn't occur, this could be the situation, which is known as incomplete lineage sorting. If they have been sorted based on this gene, I would simply say that reciprocally monophyletic, they have come from common ancestor, but they do not resemble each other in terms of haplotypes or in terms of population. So that means the level of, let me put it in different way. For example, if you sample populations of some, say, moths from different islands, and then at one place you made a phylogenetic tree, and you find that the genes which you are using, population A and B, they are mixing with each other on that tree. When you find the pattern, you say a mixed pattern, green, yellow in that particular, it means still they are sharing some kind of alleles. And in some populations from different islands, you find that this particular population of this island has purely the kind of bars which you are getting, say, green, entirely different. Lineages and yellow, entirely different. There is no intermixing. When there is no intermixing, I would say they are reciprocally monophyletic. There is a complete differentiation. If there is an intermixing, I would say they are not completely differentiated. See here, this was published in Nature 2017. So how you look at this? We say hybridization does not occur when we think in terms of Ernest Mayr. This is one species. We say mutation, then two new species. This is a typical scenario that there should be some variation in population, maybe due to mutation or some selection pressure. That population have variation, it should get adapted to a new scenario, new habitat. Then obviously we have two species. And over here, this is an objective example which has happened in cichlid fishes. Two species in the same lake. Then we have hybridization. Look at the DNA. There is mixing and there is less time. When we think in terms of speciation or in terms of species formation, we say millions of years, billions of years, but in a very less time, and the result is this. Because there are zones in that lake and they have adapted to that zone. And when this depiction is there, look at this. It clearly shows two DNAs, they have hybridized with one another. Over here also, and here also. This is ancestral, and we got two new. And this was published in Nature, and then they saw there should be some mode which is known as combined mode of speciation. And this is a typical scenario which is happening with mitochondrial DNA. You might have heard of Bolvikia, endosymbiont. It is predominantly transferred through maternal lines, and it does a lot of things in host. It creates a shallow phylogeny, or it may create a deeper phylogeny. That is, influences the level of phylogenetic inference, this endosymbiont, and it causes selective sweeps, or we call it as hitchhiking. Hitchhiking when some of the genes are carried to next generation time and again without recombination. So this parasite, you will find on internet lot of information about this parasite, and lot of work is being carried on this parasite. And this parasite, what it does, it infects, then changes the whole mode. Sperm egg incompatibility, parthenogenesis, killing, so that the insects which harbor this, they should not go for recombination. So this is another problem. Then we go for nuclear genes. Apart from mitochondrial genes, why do we look for nuclear genes? I said there should be a combined sort of approach, because we know nuclear genes are slow evolving genes. When I say slow evolving genes, as earlier I said, mitochondrial genes are fast evolving genes. Now nuclear genes are slow evolving genes. And single copy, no paralog. When a gene is there, if it is an ancestral copy, I call it as ortholog. Then at the time of speciation, there is duplication. We call it as paralog. So the resulting or duplicated genes, they undergo three different phases. Either it will perish, or it will form a new function, or it will form a subordinate function to the existing gene. So either perish, or a new function entirely, hemoglobin and myoglobin. Or it may act as subordinate to alpha and beta chains of hemoglobin, subordinate to other genes. So that duplication has played a major role in evolution. So these nuclear genes, what we target, they should be orthologs. We should not target paralogs. In case of ants, if we go for phylogeny or genetic analysis, now they are considering that we should analyze genome of males. Why males? Because they are haploid. So we should go for orthologs, and they are under selection constraint. Changes occur minimally in nuclear gene. Have you heard that gene coding for pepsin has undergone mutation in humans? There could be mutation, but that is a synonymous mutation. There could be changes, but the end product would be the same. End product, you will get the pepsin. There won't be any change in structure of protein. So that is a kind, that is, they are always under selection constraint. That's why they are slow evolving. So we should target nuclear genes also along with mitochondrial genes. Then if infection occurs, or if there is incongruency between mitochondrial and nuclear genes, we call it as mitonuclear incompatibility or discordance. This also creates problem because we know both evolve at different paces. Mitochondrial genes are influenced by Wolbachia also, introgression also. Then the result is, and you clearly know the gene which we target is related with electron transport chain, cytochrome oxidase, and most of the proteins in that are synthesized by nuclear genes. So there should be some sort of compatibility with both systems. If there is incompatibility, that becomes mitonuclear, or in terms of chloroplast, chloronuclear incompatibility or discordance. Now we are going for whole genome sequencing. When we are thinking of whole genome sequences, now there they have found that the whole genome is not entirely different, say of one species is not entirely different from other species. There are few cluster of genes which are different from each other, and we call it as islands of differentiation. That's why hybridization is occurring. Hybridization is basically compatibility of chromosomes, homologous chromosomes. If chromosomes are still homologous or still they are pairing, it means there is a compatibility, but there are certain regions which are still differentiated, which give a population its uniqueness or level of differentiation, which we, or based on which we call it as a different species. So they are basically islands of differentiation. Then, as I said earlier, now we have over 20 species concepts. Since Ernest Mayr gave one, there are different concepts which we are facing now, which should be followed. Everyone comes with a new species concept. Like this is mainly followed, Kevin DeCaro, unified species concept. So he combined all the stuff, and he said that we should look at a holistic picture. When I say holistic picture, this is the ancestral population. From here, you can easily understand. And here, the shade is changing gradually. Here also, the shade is changing gradually. So these shades basically represents the type of differentiation or the degree of differentiation which is occurring in different characters. That morphologically, in genes, in behavior, in ecology, slowly changing are occurring, and finally we have two different species which have come from common ancestor. The whole crux is that we should go for almost maximum type of information based on which we can say they are different species, or they do not. I won't say they do not interbreed. I would say there is minimal gene flow. If I say there is interbreeding, that is wrong in present context. I should say there is minimal gene flow between two populations. If there is minimal gene flow, I can say they are different from, or they have differentiated to such an extent that they should be categorized as a different species. When you say interbreeding, it's a broader term. Then this is one of my friends who works on ants. He came up with another species concept in 2020. Now what he says, gene and gene expression species concept. He says, go beyond genes. Now think in terms of Gene expression, that is proteins. Transcriptomes, that you should take into account gene expression also, genes also. Then you should think in terms of that populations have differentiated to such a level that they are different species. Then he says that, look at here, character system. Character system, he's taking it as a character. This whole phenomena, gene and gene expression, taking it as a character, he says, character systems with least risk of epigenetics. We know that gene expression can be modified by epigenetics also. So if it is being modified in terms of epigenetics, one individual can vary from other, which we call as phenotypic plasticity. So that kind of scenario is there. Then this is that stuff. Then we are going for metabarcoding as we are looking for environmental DNA. Now, how we should delimit species, given this whole backdrop, how we should look, because thinking in terms of just cataloging and thinking how many species are there, it's okay. It is okay. That is, I should not call them species as operational units. That is, there are 2 operational units, 4 operational units, or there are 5 operational units in this particular environment, or say 100 operational units. They are saying species is entirely different thing. So by having DNA sequences, you can say this much variety is there. That is fine. Maybe at the level of population or at the level of species. But saying species requires a lot of effort. So how we should go? There should be integrative taxonomy. There should be multi-score approach. That is congruence among different methods. This is important. Whatever method you are using, that method should be congruent with other method. If you are using barcoding, it should be congruent with morphology, ecology, or nuclear genes. Then, as I said in Kevin DeCuiro's unified species concept, different methods can capture different stages of speciation continuum, because speciation is a continuous process, and different methods can capture different stages, and we should have better understanding of evolutionary components or evolutionary processes than natural history. This is more important component, because nowadays we sit in labs and don't go out. So field biology is entirely missing. Sometimes if you go to the field, you will find a lot of information from where you can easily gather some evidence that these 2 populations are different, which you can't get in museum. So this.
Sorry, I had one question. What about incipient speciation? So they are not fully differentiated, but on the way. So will you put it in OTU or how?
It should be put in OTU. Then morphology, mitochondrial DNA, ecology, behavior, everything. Now this is a paper which was published in, I think, 18. They have done a lot of, given a lot of data that where they found, they have a lot of problems like morphology, how many insect taxa were identified, what was the delimitation definitive value, they found that it was correct, and what is the failure rate. So like that. And here also, you see that recent speciation, allopatric, parapatric, peripatric, sympatric, and on those columns they have given how operation priority of disciplines, that which method should be used for such kind of scenarios. So this is also mentioned, like here you can see genome, organization, then endosymbionts, population sizes, they are also important. So finally, I would say there should be holistic view about the group. There should be evolutionary thinking among traditional taxonomists. There should be a combined effort. Now we are thinking in terms of eco, evo, devo. We are combining evolutionary biology, developmental biology, and ecology. And accurate species delimitation is of paramount importance in the field of evolutionary biology, systematics, biogeography, conservation biology, and experimental. So these are the primary aims of taxonomy, are 2 names: circumscribe, describe, and classify the species. The first goal is convention, but rest are science. So this, you know how we have originated. Put in a clock, we originated 2 seconds before. So what Wilson has said, destroying a rainforest for economic gain is burning a Renaissance painting to cook a meal. So thank you. If you have any questions, please let me know.